It's my pleasure to introduce Claire Deo as today's speaker. As I guess many of you know, Claire comes from Paris in France. She's done a PhD at the École Normale Supérieure, working on photoswitchable complexes. From there, she moved on to do a postdoc in Geneva with Luc Lewis. So maybe some of you there still remember her, and where Claire mostly worked on redshifted calcium probes during her time at Geneva. After that, in 2019, we were very lucky in recruiting her to come to EMBL and set up a new group working on developing new molecular tools for biological imaging. And as maybe some of you remember, maybe the hardcore regulars of this bioimaging series, she was also one of the first presenters in 2020, so almost, I think, 5 years ago. And so back then, Claire presented really mostly about work done in Geneva, but also plans of what she wanted to do while leading a lab here at EMBL. And so we thought it would be a fantastic actually time now to give her the opportunity to now look back and share what she has done over the past 5 years here at EMBL. And I'm curious to hear how you sort of summarize the work done so far. And I'm sure there will be plenty of discussions afterwards. So with this, I hand over to you, Claire. Please take it away.
Thank you, Robert. Yeah, very excited to be back in the seminar series. It's good to see a lot of familiar faces still at Geneva. And I also remember 5 years ago when I gave the seminar, I think I was very sick and I gave a terrible talk. So I'm also very happy to be back and to give a better version of it. And I'm also excited to show you some of the work that my group has been doing over the past few years in the space of tools for imaging. So yeah, imaging has really become essential for the investigation of biological systems. And this is really thanks to a broad range of imaging modalities that can provide information across a large variety of scales that are adapted to specific biological questions and specific biological samples as well. And to reach their full potential, these modalities, they need molecular tools that can be targeted to specific biomolecules of interest and can provide a source of contrast, whether it is for the static marking of features of interest or dynamically reporting on some biochemical changes. So this has been the focus of my group's research to tackle the development of these reporters and to help these modalities reach their full potential for in turn enabling the investigation of biological systems across scales. So to address this widespread range of imaging techniques, we anchor our work in a common molecular platform for the engineering. So we exploit synthetic dyes that have many advantages. They're very bright, usually highly photostable. They can be easily functionalized to introduce different moieties in there. And they have several advantages compared to fluorescent proteins. So in particular, brightness, as I was mentioning, if you look across the entire spectral range, common commercially available synthetic dyes outperform clearly all existing monomeric fluorescent proteins across the entire spectral range. So across all the synthetic dyes available, we particularly like rhodamine dyes. They're very compatible with biological systems. They're very widely tunable, and they have this interesting property that they exist in equilibrium between an off state that is non-fluorescent and an on state that is fluorescent. And so this offers a built-in mechanism for fluorescence modulation that we can then exploit to build functional reporters based on it. And to be able to use these synthetic dyes in more complex systems, we combine them with the self-labeling proteins, which can be expressed as fusions of proteins of interest, are not fluorescent, but have the ability to recognize and spontaneously bind to a small synthetic ligand that we can attach to our variety of fluorophores to result in fluorescent labeling. And among those, we exploit primarily the halo tag, which has been one of the most studied and evolved one that works extremely well in different biological samples from cells to organism, shows very high labeling specificity, and speed is completely bioorthogonal. And so we combine synthetic chemistry on one hand with protein engineering to develop these systems that we called Chemigenetic for chemical and genetic, and build into the system new functional and optical or contrast properties. And in turn, we establish generalizable engineering approaches that can then be applied to various modalities and more broadly transferred to other tools. So With this engineering platform in hand, we address a wide set of different challenges in the imaging space. So fluorescence microscopy has remained as the forefront of our work. And while there are many excellent fluorophores out there, we're still lacking a lot of functional reporters. So we've been working on development of biosensors, for example, and light controllable tools. Deep tissue imaging has also been one key focus of my group, in particular with the development of photoacoustic probes, and more broadly, the development of chromophores that are compatible with in vivo imaging. And finally, we also aim to address some unanswered challenge in bridging imaging modalities, as well as applying them to new biological systems, which requires the identification of new contrast agents. And so today I will highlight a few of these different projects that we've been working on in my group. So first, I will tell you a bit about our efforts in the space of light controllable tools. So one of the things that we've been working on is the development of light control reporters that enable the irreversible marking of cellular targets. So you can label biomolecule of interest, and when you illuminate, you can initiate some fluorescent labeling that you can then repeat. And if you swap different fluorophores, you can do this sequentially to establish some level of optical barcoding. So this idea of just like activating permanently a fluorescent reporter is not particularly novel. This can be done with the photoactivatable fluorescent protein, for example, but you're limited to a single color. So one approach to be able to do this sequentially with multiple colors is to use photoclickable probes. So using small molecule that in this resting state is completely inactive, but upon illumination can be converted to an active state that is able to spontaneously undergo a click reaction with the fluorophore. And by performing this cycle multiple times and swapping the color, you could generate some iterative fluorescent marking with different colors. And when you combine this with the halo tag technology, you can do this in a protein-specific manner. So this was one of the ideas of Francisca and my group to pursue this. And so for the molecular system, she looked into this photo-switchable system that can photo-switch between a stable epoxide form that is non-absorbing, but also non-reactive. And upon illumination, can be transformed into this zwitterionic species that can then spontaneously undergo a 5+2 click type reaction, either with traditional clickable probes, either based on trans-cyclooctane or bisignonane type of rings to form some fluorescent adducts. And one interesting property is that the system is reversible, so you can also turn off the reactive species to temporarily control your reactivity. So Francie started investigating the switching process of this compound. And so in the dark, the species does not absorb, but upon illumination at 375 nanometer, you form the zwitterionic species, which absorbs in the visible range. And this is reversible, and you can then turn it off. So she then went on to design and synthesize the halo tag ligand based on this approach. And one of the first very interesting observation is that when the species is attached to the halo tag protein, it dramatically changes its properties. So the photo switching can still occur, but the absorbing species is blue shifted by about 100 nanometers. So this was quite surprising to us. It suggests that this compound actually interacts pretty tightly with the protein, which transforms its environment and shifts its optical properties, which might be an issue as it can prevent somehow reactivity. But it's still a fully functional photo switch in which you can turn it on and off for this reaction. So to try to minimize the interaction of the photo switch with the protein itself, Francie then looked at extending the ligand itself to push the photo switch away from the protein scaffold. And so what she found was that if she extends the length of the linker from small, which is the traditional halo tag ligand, to a slightly longer medium, to a much longer large, the optical property, the absorption actually does not change, but by putting the photo switch away of the protein, the switching efficiency actually improves, and you can form more and more of this reactive species the further away you are from the protein. So nevertheless, after that, she went on to investigate the reactivity of the species and how it would undergo click with a traditional dye. So she looked at the BCN version of JF549, of rhodamine, and she looked at this click reaction in vitro. And what she observed was that with all the different ligand tested, small, medium, or large, the click reaction actually occurs very well and exclusively in the illuminated state. So in the dark state, we observe no fluorescent labeling, and in the on state, we observe fluorescent labeling. And if we quantify this, we can actually reach labeling comparable to full saturation of the halo tag with the dye. So this was quite promising. It was all in vitro. So then she moved on to testing this actually in cells. And initially, we're looking at labeling on cell surface, which can allow us to label in different colors cells inside the more complex samples to track them over time. So for this purpose, she tested those 3 different ligands with a cell impermeable clickable dye, which was, in this case, Cy3. And so in the dark, with no switch, we see absolutely no labeling. And with the 3 of the different linker lengths, upon illumination, we observe fluorescent labeling. And on the cell surface, we found that the different ligands actually perform very similarly and result in controlled fluorescent labeling on the cell surface. So we're at this stage where Francie has demonstrated that those photoclickable ligands can enable controlled labeling of proteins on the surface of living cells. And she's now working on translating this effort into the inside of cells, which requires some more molecular engineering effort. And then we're going to work towards demonstrating multicolor and sequential labeling. And we think that to achieve this, the ability to reversibly control the reactivity to turn off unreacted species and be able to restart the reaction in a controlled fashion will be a key advantage over other type of strictly photocaged and irreversible systems. And we're also quite excited about this approach with the possibility to apply it to other things than fluorophores. So in principle, we could attach this clickable motif to a variety of other type of motif adapted to other imaging modalities, but also beyond imaging, we could imagine some oligonucleotide barcodes or affinity capture moieties that we could then extend this approach to barcoding. So in the space of light controllable reporters, we've also been working this time on reversible reporters in which we can modulate on and off multiple times the fluorescence of emitters. And this type of chromophores, they're particularly useful for different type of methods. A key one is super resolution microscopy, but can also be used for other things such as performing background correction of the signal, which is particularly useful in certain modalities. And as a design principle, these methods to modulate the emission of fluorophores can then be used and exploited for the design of light-gated reporters such as biosensors. So for this approach, Francie and Bego addressed this by developing some photoswitchable fluorophore based on a protein scaffold. So they exploited, again, rhodamine in combination with halo tag, and this approach was based on the properties of dyes to be fluorogenic. So this dye is Genialia fluor 635, and it has this interesting property that in solution, it is virtually non-fluorescent, completely shifted towards its closed form. And when it binds to the halo tag, the change of the environment around the dye and some specific interactions lead to a shift of this equilibrium, which turns on the dye. And so upon binding, we see about 100-fold of fluorescence turn on, strictly based on the change of environment. And so the reasoning was that we could build a halo tag protein that would be photoswitchable, and that upon binding to the halo tag, this chromophore will remain in the dark state, non-fluorescent. But upon illumination, the conformational change of a light-sensitive protein domain would alter the environment around the dye to create this fluorescence turn-on effect. And this is not a unique approach. We have worked on this in the past, back at Genialia in particular, to design biosensors, which I will tell you a little bit more about later on. But this was never really applied for light-based modulation. And the advantages of an approach like this is that the fluorophore itself can only become active and light responsive when it's bound to the tag itself, which avoid problems with background in principle. And the other advantages is that the fluorescence process and the light response process are completely electronically decoupled, which gives an opportunity for both high fluorescence and high photo switching properties. So to design these tools, we needed a light sensing module that would undergo a large conformational change. And so we focused on the love domain of Venus sativa, which is an extremely well-known protein domain that upon illumination undergoes an undocking and unfolding of a large helix thanks to the photocycle of an FMN cofactor into the system. And those are particularly used for optogenetic type of tools. So they've been well studied and characterized all the way to organisms. So to engineer this photo switchable halo tag, we looked at the structure of the halo tag, which is the common approach of engineering such protein, identified some positions in close proximity to the dye in flexible loops, and went into inserting the love domain into the scaffold. So we screened about 20 different type of topological arrangement early on, and we classified them based on their ability to capture the dye, because modification of the protein scaffold can completely affect how it will capture the dye, and we want to retain this property to be very efficient, but also measuring the fluorescence of the off state to see how much the dark state of the protein would already open the dye. So this is all, in the dark state with no light, and we can see that all the way up here we have halo tag, which captures the dye extremely fast and shows very high fluorescence. And on the other hand, we can see the love domain alone, which does not capture the dye at all and does not have any fluorescence if it does not capture the dye. And so we're able to rank all these constructs across the board. And we focused then primarily on the ones that did retain efficient capture of the dye. Some of them did open the dye very efficiently, similarly to halo tag, and some of them opened the dye very low, which was quite promising for our approach. And when we looked at this different constructs and illuminated them, we found that 2 of them actually already displayed some modulation of their optical properties based on the illumination. So in particular, this construct, which is the insertion at position 143 of the love domain, showed a turn off in absorption of about 50% upon illumination. And 154 also showed similar effect, but it was already almost fully open in the dark state. So we focused on this 143 construct as a starting point, thinking that we could engineer the reverse process to access very large turn on based on this. So Franci and Bego then undertook some sequential site saturation mutagenesis rounds on this initial construct. And starting from this negative going variant, they could rapidly isolate variants that showed the large positive turn on of fluorescence upon illumination. And so after a few rounds of mutagenesis, we isolated a top construct that we very originally called PS halo tag for photo switchable halo tag. And so we characterized this protein. We can see that when we bind it to the far red 635 dye, upon illumination, we get close to a 5-fold turn on, both in absorption and fluorescence, which demonstrate that our system actually works by tuning this open-close equilibrium, modulating the absorption of the dye. And this is reversible. The love domain is thermally reversible. So if you illuminate and then you let it sit in the dark, you can repeat this several times. So we solved the crystal structure of this protein bound to the fluorophore. And what we could see that's interesting is that this long helix of the love domain that is responsible for the conformational modulation in those love-based protein, it actually becomes fused to one of the top helix of the halo tag. And so we think that this contributes to the mechanism of modulation hereby, where the helix undocks and unfolds upon illumination, the conformational change is propagated to this top helix on the halo tag. So what can we do with this? We can, of course, do some further engineering to refine the properties and tune the properties. So one interesting thing is that we can take advantage of the knowledge that exists on the love domain kinetics, and that we can introduce point mutations to tweak the kinetics of both the turn-on and the thermal turn-off relaxation, which remain not very fast. And we believe it's because we do have to rearrange conformationally the entire protein, but we can substantially speed up the process in both ways by introducing point mutations without compromising so much on dynamic range. And one other thing that we can do, and that is like a very interesting properties of this type of chemigenetic system, is that with a single protein, you can swap the dye to fine-tune the different properties. And so if we look at JF635, if we replace it with closely resembling fluorophores, but that have slightly different open-close equilibrium, we can tune the properties of the system, achieving combined adducts that have either lower overall signal, lower brightness, but larger turn-on without changing the optical properties. So then we tested this PS halo tag inside living cells. We looked at different targets and across H2B, TOM20 mitochondria, and life act for actin, we can see that illumination results in large fluorescence turn-on. And if we quantify this, it's actually very conserved across the different target tested, which shows that the behavior is actually quite robust, independent of the cellular environment around it. And we can do this, repeat this cycle multiple times with losing minimal performance over 20 cycles of on-off switching. And the tunability that I was mentioning by changing the dye ligand is also retained inside living cells. So this is using a slightly different fluorophores on the same target, we can obtain higher dynamic range. And so one advantage of the system is that we can do really spatiotemporally controlled activation of cells. So if we look here at living cells that are labeled with the photoswitchable halo tag, we can select individual cells that we can turn on sequentially. So we can turn on cell 1 exclusively, then cell 2, cell 3, etc. So we can really do targeted activation of cells to track them. And as opposed to the system as I was previously showing, which is an irreversible fluorescence marking, here the system terminally reverts. So we can see that over time, this cell that was activated first, then starts slowly relaxing and reaches back the reset state. So there is a reset time. So this is mostly for short-term labeling, although we can use the kinetics tuning to play a little bit on this aspect too. And so, yeah, finally, what can we do with that? And I mentioned that one of the key applications of light responsive system is super resolution microscopy methods. So we're early on interested in the possibility to do single molecule localization microscopy with those constructs. And the reality is that the current variant that we have, which is a first generation tool, has some limitation. The dynamic range is not as high as what we can see with photoactivatable type of proteins. It is fairly good for a photoswitchable system, but the kinetics are still a limitation. The system is quite slow due to this conformational rearrangement. And so one approach that we envision to be able to make use of those in single molecule imaging is to actually exploit this photoswitchable halo tag to, in combination with dyes that have interesting blinking properties, so that in the dark, the dyes would be non-blinking, and that upon illumination, it would promote fluorophores into a state where they would spontaneously blink. And so why this is interesting compared to regular spontaneously blinking dyes is that this provides a mechanism to modulate the amount of emitters that can help to repopulate a decent number of emitters for imaging throughout the acquisition, and therefore prolong the acquisition despite bleaching and relaxation. So you can pump back emitters in the on state. And so this is very preliminary and recent work with our colleagues at the imaging center. But this is so in living cells. If we look at H2B tagged with our photoswitchable halo tag, in the dark we can have a very low number of emitters, and then upon illumination we can start pumping back up the number of emitters overall. That is then retained after illumination for some amount of time before it relaxes. So on this part, what we've developed is actually a new mechanism for fluorescence photo switching based on this conformational modulation, which has not really been looked at before. We can achieve up to close to 10-fold turn on in vitro with the best combination of fluorophore and protein. We found that the performance in living cells is quite robust, and we can demonstrate that this can have some interesting application in live cell single molecule imaging, although there's still some work to be done in that space. Sorry. Okay, so now I will tell you a bit more about the work that we've done on the development of probe for deep tissue imaging, which has also been a strong focus of interest in my group since my time at Genelia, where I was working on far-red chromophores. And so we're interested in probes for deep tissue imaging, and particularly for neuroimaging, to be able to do this all the way to the mouse brain. So one challenge is that fluorescence microscopy in vivo is limited to about a couple of millimeters in depth, which is due to light scattering. So using fluorescence, we can achieve very high resolution imaging of activity of single neurons, but only of a small field of view and at relatively superficial depths in the mouse brain. So to be able to image non-invasively deep in tissue, we need new methods. And for this, photoacoustic imaging has been really a key focus in our group. So this photoacoustic imaging is an emerging imaging modality which relies on the detection of acoustic waves followed by excitation using light. So the advantage is that this method can achieve much greater imaging depth compared to fluorescence, so which is particularly promising for in vivo imaging of very large field of view, albeit at a lower resolution of about 30 microns. So we're in this space, we're particularly excited about the idea of realizing imaging of the neuronal activity across the entire mouse brain. So this has been the focus of a very close collaboration with the Prevdal group in the past few years, who have been developing some advanced photoacoustic instrumentation, while my group has been working on the probe side. So photoacoustic imaging, very briefly, how does it work? So it relies on the absorption of light by chromophore, which reach an excited state, and they lose this energy through non-radiative relaxation, which results in a temperature rise that is propagated as acoustic waves that can then be detected and reconstructed to rebuild an image. So one of the key challenges is that this effect of absorbing and then relaxing with non-radiative relaxation is what most endogenous molecules actually do. So they provide a very large source of background, which is problematic for this modality, in particular blood and hemoglobin. And so one requirement is to really have probes that work in this near-infrared window where the absorption of this endogenous chromophores become minimal. So the general principle of this modality actually explicits quite well what are the requirements to build very good photoacoustic reporters. So on the optical side, those needs to have very high near-infrared absorption. So in this window to avoid endogenous chromophores. They need to have a very low fluorescence quantum yield. They need to be very terrible fluorophores to maximize this non-radiative relaxation. And they need to have very high photostability because photoacoustic requires a very high energy pulse laser. And then there's the general requirements that apply pretty much to all type of reporter. They need to be compatible with specific cellular, subcellular labeling. They need to be usable in vivo, which is the main space where we would want to use this modality. And then they need to be able to be engineered to generate biosensors. And in particular here, we're thinking primarily about calcium biosensors to be able to visualize neural activity. And so this has been the use of existing calcium biosensors for photoacoustic imaging has been looked at before. So the most popular calcium sensors, GCAMs, have been tried and demonstrated in photoacoustic imaging. So what you can see here is inside the mouse brain in vivo through the intact skull. And you can image GCAM, and you can even visualize some transient of activity in response to stimulation. But the issue is that GCAM is green. And so there's a problem of penetration. So you can only visualize it using photoacoustic at this very superficial depth. So the wavelengths here is really 1 key limitation, although the sensitivity is very good. The other aspect is that GCamp has been heavily optimized for fluorescent. So in contrast, the photoacoustic signal that you get from it is relatively small. On the other hand, you have more recent near-infrared protein-based calcium indicators, such as NearGecko, which has the advantage to be really red-shifted exactly in this absorption window where we would need it. One of the challenges that this near-infrared protein-based sensors, they have a relatively low sensitivity, and those NearGeckos, they only work in turn-off mode in response to calcium with a relatively small turn-on. And this is very recent work from this year, where one of the more recent iteration of NearGecko was actually looked at in photoacoustic in vivo, and they were able to demonstrate that they could detect NearGecko and find out where it is and separate it from the blood background. But they were not able to see calcium dynamics based on it, which could be due either to the lower sensitivity of these probes or to the very high bleaching rate that they have under photoacoustic excitation. And so we reasoned that a promising approach to generate photoacoustic calcium reporters could be to exploit this chemigenetic type of system in which we could really engineer the chromophore and the protein to create such sensor. And the reason behind that was work that I contributed to back at Genialia, where we developed a far-red calcium sensor based on this chemigenetic approach, in which very similarly to our for the switchable halo tag that I just presented, we had introduced a conformational-based calcium sensing domain in which, upon sensing calcium, would undergo this large change that would modulate the fluorescence of a rhodamine dye bound. And this sensor displayed actually very nice properties in vitro for a far-red calcium sensor with absorption in the right area and very good sensitivity. However, this sensor was originally optimized really for fluorescence, and so it is extremely bright. And so we set out to re-engineer this for photoacoustic by reasoning that we could take this open-close equilibrium of this fluorogenic dyes that goes between this state that does not absorb and this state that absorbs and fluoresce, and convert this dye into acoustogenic dye that would, in one state, still have no absorption and therefore no photoacoustic signal, but in the open state will have a very high absorption, but no fluorescence at all, which would guarantee a very high photoacoustic signal. So based on this consideration, we rationally design a set of target dyes combining substitutions for red shift, quenching group, and then tuning of this equilibrium to position it in exactly the right space to get large turn on. And so this was really the work of Alex, a PhD student in my group, and it was actually a pretty challenging synthetic effort. And Alex started to look at different families of dyes rationally designed based on this different consideration. And for this, we also really have to thank the Levis group and John, who actually had already made some of those that they gave to us to test in our type of assays and approaches. So I will spare you the chemistry, but Alex basically looked at 3 key families of dyes. So some silicon rhodamines, some rhodamines substituted by perl that had the property to extend the electronic conjugation to shift them toward the red while lowering the fluorescence quantum yield. And then some malachite green derived dyes, which malachite green is actually a very old chromophore that had really not been looked at in this context. And so he did some different synthetic modification and systematically evaluated those dyes based on their absorption wavelengths, which for us is very critical that we work above 650 nanometers, and looking at their equilibrium. So we want to avoid dyes that are either too open or dyes that are too closed that would not lead to this modulation that we want. And so based on this, he selected a panel of dyes to then synthesize the halo tag ligands and start looking at them using actual photoacoustic. So so a small panel of the top candidates that he found was this malachite green series with different type of substitution. And we initially looked at their absorption property, but absorption is only a proxy for photoacoustic imaging. So we needed to move towards actual photoacoustic characterization, which is not so straightforward because you cannot get commercially available photoacoustic spectrometers. So we worked closely with Nikita in the Prevedel group, who designed a multimodal photoacoustic spectrometer, which requires that the cuvette is inside a water bath for acoustic coupling. So it was a lot of work, but it works very nicely. And this allowed us to do different type of measurements. So first, we found that those ligands, they all display a large photoacoustic turn-on upon binding to the halo tag, which is what we wanted, and that this property actually matched really well with the absorption property. We looked at photoacoustic signal over time, and we found that once bound to the halo tag, this chemigenetic systems were much more photostable than synthetic dyes and fluorescent proteins that were spectrally matched. And we also looked a bit more closely at the rationale behind the fluorescence quenching of our approach, and we compared the original dye of this chemigenetic calcium sensor, Genialia fluor 635, which, as I mentioned, is extremely bright, 75% quantum yield. And we compared it to this malachite green, which is 0.1% quantum yield. And so when you bind it to the halo tag, they actually have exactly the same absorption. But when you look at the photoacoustic signal, the malachite green results in about 17-fold more signal based on really this quenching effect, which is a key requirement to achieve this large photoacoustic signal. So this is all using those dye ligands as halo tag labels, so really as static markers. But based on this, we can also generate a series of calcium sensors for photoacoustic imaging. And for this, we simply combine them with the original HaloChemp calcium sensor protein without any modification. And here again, we see the power of the tunability of this chemigenetic platform, in which we're able to translate this design to a new modality simply by swapping the dye ligand. So this is just an example of in vitro characterization of the sensitivity of the different calcium sensor. So we have two variants of this HaloChem protein, 1A and 1B. And together with our 3 top dye ligands, we can generate this suite of different calcium indicators with tunable properties. Some of them provide slightly more signal, some of them slightly more turn on. And we compare them to this fluorescent protein based near gecko-1, roughly at the same wavelengths. And we can see that in terms of sensitivity, those sensors clearly outperform near gecko, which only shows, as I was mentioning, a relatively small negative photoacoustic turn-on. So this is in vitro in cuvette. We can then start looking at the performance of the sensors in more realistic conditions. Looking at them in what's called tissue mimicking phantoms, which is an in vitro system, but embedded into a scattering solution that mimics the level of scattering that you find inside the mouse brain. And that is also imaged directly on the photoacoustic tomography setup, which is the final imaging instrument to be used in animals. So if we embed this system in this tissue mimicking phantoms, we can see that we actually recapitulate extremely well the in vitro measurements, with near gecko here showing a negative response to calcium. And then this is with dye number 1, 1A and 1B, which shows a turn on in response to calcium, and 1B showing a higher response. So we see that we can image them in conditions mimicking the mouse brain, where we see some decent photoacoustic turn on. And so then we move to looking at their performance in mouse brain tissues. So those malachite green dyes, so as I mentioned, they have 0.1% quantum yield, but in fixed samples, you can have very long exposure, so you can actually see them. It's not great, but you can use fluorescence still as some level of validation. And so here what we did is that that was with the help of Juan and Gretel in the Prevedel group. So they injected mice with the halotag virus expressing in neurons tagged with the GFP. And after expression, they labeled the mouse brain by intracerebroventricular injections in vivo before excising the brain to do quantification analysis. So what we can see is that the dye delivered in vivo actually goes to the brain and can label neurons in the brain, where we find good colocalization with the halotag. And we can also look at the entire excised brain labeled using photoacoustic And where we can see that where the halo tag is expressed, we can also observe a large and specific photoacoustic signal that originates from the dye that we used, that we can also validate in fluorescence. Here you can see on the slices the correspondence with the photoacoustic signal. So this is early days. It's also a first generation of tools. We think there's still some improvement to be done. And one key challenge here, which is a reality with this chemigenetic system, is the delivery of the dye and converging to dyes that would be more bioavailable and that can be delivered easier in living system. Because as you can see here, we achieve in vivo labeling, but not homogeneously across the entire brain. But then nevertheless, so here we have achieved actually the first example of this chemigenetic approach for photoacoustic imaging. And in vitro, they have the best performance really of all photoacoustic calcium indicators that have been reported to date. And yeah, there's still some improvement to be made to reach this functional in vivo imaging. But we think this is a very promising step towards this. And so, yeah, just in the last few minutes, I want to switch gears a little bit and tell you about something else that we've been working on. So the projects that I have just presented were all very grounded in rational design, exploiting existing prior knowledge to tailor this chromophores to specific application. But the reality is that this is not always possible. And in many instances, we do not have the knowledge to build the right probes rationally. So for example, fluorescence labeling can still be an issue in many different type of samples, whether it is patient isolates, ecological samples, non-model organisms, where there can be a lack of antibody, no possibility for genetic targeting. And in addition, we see emergence of more and more new imaging modalities, which require new contrast agents. So ultimately, this requires some level of screening to identify suitable scaffold and that we can then build structural properties relationship on. So for this purpose, we've curated a library of chromophores of around 500 compounds that combine both agnostic dyes as well as ligand, that we aim to become some kind of universal resource that people can access and use to screen for their new methods or newer sample. And so we've built this library, of course, based on many different considerations. So one of them was structural diversity. So it's both based on homemade and commercially available compounds. We wanted to represent pretty much all common classes of fluorophores and chromophores to date. And the second consideration is really functionalization. So the general idea behind it is that we can screen using this library in different methods and modalities, evaluate this, and then find some heads that we can then refine and optimize using synthetic chemistry to then feed back again into this library resources. And the general idea is really to repurpose existing dyes in light of new modalities and new biological questions, which really built into also the general interests here at EMBL. And just to highlight, and so ultimately this library resource is going to be something that is hosted here by the Chemical Synthesis Core Facility, and so that is accessible to you if you want to use it. So yeah, I just wanted to show a few examples of what we're currently using this library for that we're quite excited about. So one first thing, this is actually quite older work that was published this year, and that was a much smaller scale initiative where we, in collaboration with De Virger in the Lippincott-Schwarz lab at Genelia, we tested a small library of fluorophores to identify stains for guanine crystal in zebrafish iridophores, and we're able to find dyes that can allow to track those guanine crystals over time to visualize them. Another ongoing effort is the screening and search of markers for Plasmodium falciparum research in collaboration with our colleague Gautam Day here at EMBL and some collaborators downtown, which was funded by the Health and Life Science Alliance here that also contributed to the financing the buildup of this library. And on the other hand, one thing that we're also very excited about is to use this library to identify new stains and contrasts for correlative imaging methods. In the context of the EC-funded imaging projects, we're currently screening and looking at contrast agents for correlative fluorescence and X-ray imaging that we can screen using benchtop X-ray methods. Another approach is the identification of fluorophores that can provide contrast in cryogenic conditions to build new cryo-super resolution microscopy methods that we're pursuing also in collaboration with the Zimmerman group here at EMBL. So just to conclude, in what I've shown you today, I've shown you on one hand, some projects that are very grounded in rational molecular design, where we've built some photocontrollable reporters, deep tissue imaging probes that are, generally this Chemigenetic system, our first generation probes, where we go all the way to demonstrating usability in biological sample and system, and that we hope will generate, broadly applicable tools that are more and more robust with further engineering. And on the other hand, other type of approaches that involve large-scale, more agnostic screening applied to non-model biological samples, emerging modalities, correlative approaches, to in turn help build this structure properties relationship that will feed into rational molecular design. And together, we strive to have this build and inform technological advances to enable new biological applications. So with that, I will, of course, like to finish by thanking, first of all, my group. I've presented work that pretty much everyone in these pictures has contributed to over the years, and they've done amazing work, and our funders that have made this possible, and all our collaborators inside, outside, that make all of this possible. So thank you very much.
Thank you, Claire. Maybe can I get here so that everybody hears me online? So really impressive talk. It's quite exciting to see how far it has come in the last couple of years. So we have time for questions, and maybe for those online, just quickly raise your hand so I can see you, and then you can speak up, or otherwise here in the room, just raise your hand. Please, yeah.
Comment on the time scales of the calcium indicators, because you are imaging neurons in the brain, and I assume that you want to see the activity of the neurons in the brain with these calcium indicators, but is the photoacoustic signal generation fast enough to actually see these neuronal spikes?
Yeah, so the limitation is mostly going to be the sensor response to calcium. So we've looked at kinetics using stop flow. It's very reasonable to be able to see calcium transients. They're actually relatively similar to the original fluorescent one. I do not have the numbers in mind. But yeah, we do not believe that the kinetics of the sensors itself are going to be a key limitation for this. But, I think there's also consideration in the achievable speed of the photoacoustic setup by itself, which will be another type of limit. And also the idea that photoacoustic is not a single cell resolution method. So each signal that you will see will originate for a few hundreds of cells. And so also timing, depending on the time of these transients, will be another challenge in the kinetics overall.
Luke, you have your hand raised.
Hi, Claire. Hopefully you can hear me. I'm on the Oregon coast, so it's really early here. But great talk.
Nice.
Yeah. This is not it's a real background. But anyway, so I had a question about the photo click reaction. So when that thing is bound to the protein, it has a different absorbance. Do you think the thing is protonated, or do you have any ideas about why you get that absorption shift?
We don't know. We haven't looked at it fully. We also haven't looked at pKa so much, bound and unbound. Originally, we were thinking that it could actually just be that one of the phenol is completely unconjugated due to some weird twisting, and therefore because 100 nanometer is kind of big. So it could be that, but we're also a bit surprised that, the more we push it out, we don't change ink. And on SnapTag, we see the exact same effect as well. And Francie has been looking also at more substitution on the phenol rings to really balance electronics, some very electron withdrawing group. And I think across the board, she sees the same effect.
That's a really cool observation. So thanks so much.
Thanks.
Any more questions? Kevin?
Yeah. So very nice, very cool. It kind of got me thinking, what would be the ideal photoacoustic molecule for something like this? And so the excited state lifetime malachite green still has a significant radiative component. So that's going to quench your photoacoustic signal. You could go towards all quenchers, or essentially something that has no fluorescence whatsoever. But I mean, you'd essentially then have to modify the absorption to create this like modulatable signal.
Yes. So we need to have this like absorption driven mechanism basically to get to this modulation. It's really absorption based. Yeah, I think we could go further. I think there was a series of compromises that needed to be made. One of them being really kind of like downstream usability in vivo. So we don't want to go towards this ultra conjugated quenchers either. Because it's a challenge. Malachite green, the quenching is all this rotation stuff. It causes problem with solubility, and so it's a little bit of a compromise. And I think the other reason is that initially we wanted to stay as close as possible to this original HaloCamp building blocks to not completely break function. So really going like incrementally like this.
It's very cool.
We think photoacoustic signal itself might not be the biggest problem in the end. We would like a bit more.
Yeah.
Sensitivity might be still a bit more challenging early on.
Exactly.
Mm.
And bioavailability.
Yes, bioavailability.
Okay, we have time
Claire, that was really super cool.
Thanks, Brian.
I have a question about your photo-switchable halotag fusion molecule. Did you expect that the alpha helix was going to be shared? Was that designed, or is that? I mean, it's super cool, no? But did you just see that in the crystal structure?
No, that was a little bit serendipitous that we did some AlphaFold modeling quite heavily early on, and we found that the crystal structure actually matches very nicely on this thing. But it was interesting. We had this early hypothesis. So when we modeled this very first variant that was negative turn on, then we had in the process some variants with very small turn on. And when we would model the structure, we would not see this helix conjugation. So we kind of assumed that this was kind of like a requirement to get this large turn on. But based on all the variants that Francie and Bego have looked at in screen, I'm not so sure it's like very binary like this. It can be more subtle. Yeah, and I think the other challenge is that which is the same as for everything that has two states, is that we actually have zero knowledge on what the other state looks like. So it's kind of, we actually don't know if in the on state we have like a fully unfolded thing, or if it's just like a tiny movement that somehow changes everything.
More questions? If not, maybe I have one also on the photo switchable halotag. So you've used that for, or you want to use that for super resolution imaging. And so I was wondering, have you characterized a little bit, like, of course, you're switching on and you're emitting more photons? Like, have you somehow characterized how many photons you get emitted? And because that, of course, influences the achievable resolution.
Yeah, so I mean, Cariel is the one that has looked at that. It's also, very preliminary results. And it's actually, we actually don't switch the number of photons. The number of photons is actually constant. We just switch the number of emitters, but each one of them has the same number of photon in the off and on state. So this doesn't change so much. Yeah, I think, same thing. We'd like a bit more photons down the line, but it's hard to go brighter than the JF dies. So I think we'll need to do with what we have.
Thanks.